So this is an oxidation reaction. The next step is an enzyme called fumarase. It's a hydration reaction. So we're starting with fumarate that had a double bond between carbon 2 and 3, adding a water across that double bond to make this 4-carbon compound malate. Malate has a hydroxyl group at this position that came from the water molecule. The next step, malate dehydrogenase, oxidizes that malate and oxidizes that hydroxyl group to form a ketone, and the electrons are transferred to NAD to form NADH. So it's another step that produces an NADH that can be used as an energy source later on for making ATP. And now we're back to the 4-carbon molecule we started with, oxaloacetate. So that completes the cycle. So 8 reactions in the citric acid cycle. So this is the citric acid cycle. What I encourage you to do is to draw this yourself, write the structures, write the names of the enzymes, be sure you know which steps produce NADH and CO2, FADH2, this ATP step, and know the structures of the intermediates. If it was a smaller class, I would be able to ask you to, on a test to draw those structures and write the pathway, but since it's a big class, we have to use Scantron, so what I'll do is show you a structure and ask you some question about it. And so you should be able to recognize, oh, that's malate, and what it does, where it is in the pathway. And as I said when we talked about glycolysis, I think using a pencil and writing this down yourself is a good way to help you learn it, much better than just looking at a screen or a book. The actual writing and drawing affects your brain and makes you learn it better. So as we look at this pathway, this is a little diagram to think about what is the fate of the carbons. So we have 2 carbons from acetyl-CoA, 4 carbons from oxaloacetate. So in this drawing, the acetyl-CoA carbons are pink and the oxaloacetate carbons are black. So we form citrate and isocitrate. We release 2 carbon dioxides. The 2 carbon dioxides that are released came from oxaloacetate, not from acetyl-CoA. So as we get down to the 4-carbon compound, we have 2 carbons that came from the original oxaloacetate and 2 carbons that came from acetyl-CoA. And so that keeps going around the cycle. And each time, that's the fate of those carbons. The carbons that are released as CO2 are carbons that were originally in oxaloacetate. Okay. This is a molecule called malonate. Malonate is a competitive inhibitor of succinate dehydrogenase. So it binds in the active site of succinate dehydrogenase and inhibits it from carrying out its reaction. So it fits well in the active site. So I want you to think about, why is it an inhibitor and not a substrate for succinate dehydrogenase? And then what happens to the citric acid cycle intermediates, the compounds, if we add malonate to mitochondria that contain the citric acid cycle enzymes? What is going to happen to the concentrations of those molecules? So talk to each other about these questions for a few minutes. Okay, why is malinate not a substrate of succinate dehydrogenase, but instead an inhibitor? Here's this reaction, succinate dehydrogenase. So malinate inhibits this step. Transferring electrons from succinate to FAD.
There's no hydrogen left.
Right, there's no hydrogens, so these two carbons in succinate are methylene groups, so CH2. And if we look at succinate, there's no such thing that can be used as an electron donor to reduce that molecule. So it fits in the active site, it binds there tightly, it competes with the substrate, but it can't carry out a reaction. So it's just stuck there as a competitive inhibitor. Someone mentioned that if you had a high enough concentration of the substrate, succinate, we could outcompete the inhibitor. But in cells, if you add this malinate to it, cells usually there's not high enough concentration of succinate. But what happens if we inhibit succinate dehydrogenase? What happens to the concentration of succinate? It's going to increase because it can't be used. It can't go forward in this reaction. So we'll see a buildup of the concentration of succinate and then successively buildup of everything before that because everything's backed up like a traffic jam because there's a block here at the succinate dehydrogenase step. And there'll be lower concentrations than these molecules that are after succinate dehydrogenase in the pathway. Okay, so these reactions are regulated. Pyruvate dehydrogenase complex and the citric acid cycle are regulated in a coordinated way. And so pyruvate, I mentioned, can be used to make acetyl-CoA. It can also be used to make glucose in gluconeogenesis that we talked about on Wednesday. It can be used to make amino acids. So how pyruvate is used is controlled by the cell, depending on what the cell needs. If it needs energy, then pyruvate dehydrogenase is going to decarboxylate it to make acetyl-CoA to go through the citric acid cycle. If you've got enough energy, then that acetyl-CoA can also be funneled off and used to make fatty acids, to make lipids to store. So if you eat a lot of sugar, you convert it to acetyl-CoA, that can be used to make lipids. The production of acetyl-CoA is a committed step, so we can't go backwards in this reaction from acetyl-CoA to pyruvate. At least plants can do that, but animals can't. So once acetyl-CoA is made, it can't be used to make sugar, for example. So let's look at the regulation. We already talked some about regulation of pyruvate dehydrogenase complex. It's inhibited by ATP and acetyl-CoA and NADH. Its own products inhibit it. And ADP and pyruvate activate pyruvate dehydrogenase complex. In the citric acid cycle, there are two key regulatory enzymes. It's the two enzymes that carry out oxidative decarboxylation steps. So isocitrate dehydrogenase is inhibited by ATP and NADH and activated by ADP, and the alpha ketoglutarate dehydrogenase is inhibited by ATP by its own product succinyl-CoA and by NADH, also one of its products. So these are the key regulatory steps in the citric acid cycle, and they're coordinated with pyruvate dehydrogenase regulatory steps to regulate the flow of atoms through the citric acid cycle. The citric acid cycle is connected to gluconeogenesis through oxaloacetate. We mentioned already. There's the intermediates in citric acid cycle can be used in other kinds of anabolic pathways. and to make amino acids, for example. And so we call a pathway that's both catabolic and anabolic, that's an amphibolic pathway. So the citric acid cycle has both catabolic and anabolic functions. It's an amphibolic pathway. And we're going to look at what some of those intermediates can be used for. Here's the little diagram of the pathway. Oxaloacetate, the starting point, is made from pyruvate. It can be used then to make glucose through gluconeogenesis. Oxaloacetate can also be used to make the amino acid aspartate, which is a starting product for making some other amino acids and some nucleotide bases, purines and pyrimidines. Succinyl-CoA is a precursor for making porphyrins. So heme and chlorophyll are porphyrins that are made using carbons from succinyl-CoA. Alpha-ketoglutarate is a precursor for making glutamic acid, and glutamic acid is involved in synthesis of other amino acids and purines. Citrate is used for making fatty acids and some sterols. So there are connections between citric acid cycle and lots of other metabolic pathways. Okay, so coordination of citric acid cycle is linked to coordination of glycolysis. So glycolysis is inhibited by ATP and NADH. Phosphofruct, which are products of the citric acid cycle. So citric acid cycle products are regulating glycolysis. What's the most important enzyme in regulation of glycolysis? Phosphofructokinase, right? The third enzyme in glycolysis is this most important regulatory step. It's inhibited by ATP and NADH. It's also inhibited by citrate. So that's a connection between citric acid cycle and glycolysis. So the most important regulatory step in glycolysis is inhibited by the first product of citric acid cycle. When citric acid concentration is high, cells don't need to do so much glycolysis. The pyruvate dehydrogenase complex is inhibited by NADH and acetyl-CoA, its own products. And so that helps to regulate the flow of carbons from pyruvate through to carbon dioxide. Here's the net reaction of the citric acid cycle. So we're starting with one acetyl-CoA, oxidizing those carbons to 2 carbon dioxides. We start with 3 NADHs and reduce them to 3 NADs, reduce them to NADH. 1 FAD reduced to FADH2. We phosphorylate a GDP to GTP and release a coenzyme A. These electrons in FADH2 are transferred to a cofactor called ubiquinone, and that ran off the bottom of my slide somehow. I'll show you that name again in a minute. So this molecule is ubiquinone. That has this 6-membered ring with 2 carbonyl groups on it. FADH2 transfers its electrons to ubiquinone to make ubiquinol, and that is an important intermediate in how those electrons are moved, and we'll talk about that later too. I think we will stop there.